Here at the Nature Files, we know that environmental DNA can be used to solve all sorts of cases through the powers of detection. That is, detecting different species in the water, which has a myriad of applications, from assessing ecosystem health to studying individual species. But this type of presence or absence detection is just the beginning of what eDNA might be capable of. Our next case pushes eDNA into more unknown waters, taking a deep dive into the world of juvenile salmon to test new advances in eDNA technology. Join us as we crack the code on nature and unlock mysteries in The Nature Files.
So we're in Okasolo Channel, and we're here because we do quite a bit of seining here. This has been one of our most productive kind of sites throughout the years.
A salmon's migratory life cycle takes it through all sorts of habitats as it makes the journey from the ocean back to its natal river to spawn. By improving the tools we have to learn about salmon, we peer through a window to our entire ecosystem's well-being.
So I think that their life history, it really lends itself to kind of understanding multiple impacts of what's happening with climate change or with habitat quality.
For years, Brett Johnson has been working with juvenile salmon to find out their migration patterns and where they're spending time in the nearshore environment, learning about what factors affect their survival when they are at their most vulnerable. And he's doing it using environmental DNA. This week, Brett and his team have organized an eDNA extravaganza to test if this technology can be pushed past questions of mere presence or absence.
My project is hoping to go beyond that and look to see if we can correlate the amount of DNA that we're getting in the water with the amount of fish swimming through.
If Brett and Natalie succeed, and these methods prove to be credible, it would open up a whole new world for eDNA. And for the salmon research community, it would lead to better, more accurate studies of these vulnerable juveniles. First up, Brett and Natalie will be sampling for juvenile salmon at a number of different locations and depths. At each sampling site, the team will use eDNA along with a more traditional method of sampling salmon, seining. By cross-referencing those two sets of findings with visual observations, they'll be able to determine if eDNA is reliable at measuring not just the presence of different salmon species, but also their relative abundance. If so, eDNA could serve as a supplementary method to seining, offering new insights and potentially mitigating some of its biases.
There's this guy. See this one?
This approach is also more cost-effective and less harmful to fish, addressing some of seining's drawbacks.
And that's kind of one reason my project, using environmental DNA, came onto the scene, to kind of not only see what we are catching, but also what we're not catching. And so by using a genetic method such as environmental DNA, you're capturing not only fish that are biased to be captured in the net, but perhaps fish that are swimming a little lower, or ones that are too big to fit in the net, or too small and would swim through the net.
Another perk of eDNA? We can collect samples deeper than any of these seine nets or even divers could go, allowing the team to look at what they call fine-scale distribution patterns of salmon, finding out exactly what habitat these fish use as they migrate, which will help the researchers to better study these juveniles in the future.
We're heading out to our second site, which is a little further offshore, and we're going to be doing 3 depths there. So 0 meters, 30 meters, and then 5 meters off bottom. It'll be about 100 meters or so.
But how exactly do they get a scoop of water from 100 meters below the surface? To do that, the researchers have bottles that are open at both ends as they follow a track down to depth, at which point a trigger prompts the bottle to snap shut. After gathering 24 water samples during their eDNA extravaganza, the team packs up their samples and heads home to get ready for the next step: analysis. Now, when it comes to using eDNA to measure abundance, the key is finding a way to focus in on the DNA of a single species. To do that, you need a tool called quantitative PCR, or qPCR. Unlike other eDNA methods that resolve whole communities of organisms, qPCR focuses on a single species and can tell you how much DNA from that species is present. Natalie has spent months getting these species-specific eDNA tools working. Now that she's got samples in hand ready for analysis, she's hoping that qPCR will prove to be accurate in estimating not just the presence of salmon species, but the size of each species of salmon's populations here.
It's quite challenging to estimate abundance from eDNA. I'm beginning to get an idea of certain abundances from our captures, but it is quite challenging to have one model that really incorporates everything, because we work in really challenging ecosystems where there's tides and currents and biotic and abiotic factors all contributing to the input and degradation of DNA in the water.
After many months of working to separate out all the Pacific salmon DNA from their samples, the team is finally starting to get some results. Their seining and eDNA samples show that pink salmon appear to be widely distributed in their sampling area, occurring at all depths and distances from shore, whereas chum salmon appear concentrated in the surface waters. Knowing this will help researchers more accurately sample and study these salmon moving forward. By comparing their eDNA findings to the seine readings, they've been able to determine the best tools to isolate the DNA from a single species, meaning they are one step closer to being able to rely on eDNA for abundance estimates. That's a huge stride forward for science, and a case closed for the scattered salmon. So, should we crack open our next case file? Next week on the Nature Files, we explore the case of the colonizing crabs and find out how researchers are using eDNA to stop the spread of the invasive European green crab.